Hello and welcome back to lecture 10.2, the cell cycle. Phases of the cell cycle. Specifically, we'll be talking about interphase and M-phase. Before that though, let's talk about lecture question 10.1, compare and contrast which of the following are true about chromosomes. What do you guys think? If you said A, you're correct. Nicely done. Okay, let's move on to lecture 10.2. Okay, so here's where we left off in lecture 10.1 with the cell cycle of eukaryotes. If we remember from before, the cycle is split into two stages, interphase and M, or mitotic phase. Interphase is this kind of whole gray bit here, and M phase is just this tiny little sliver here in orange? Yeah, I guess it's orange. Anyway, most appropriately, we'll be starting our conversation by talking about the biggest piece of this whole pie, interphase. Of course, interphase consists of 90% of a cell's life cycle. And we can break it up into three steps. G1, or first gap, or gap 1, I guess. S, or synthesis. And G2, or second gap. Now, the cell grows during all three of these steps by adding phospholipids to its membrane, making extra ribosome, making its organelles bigger so they're ready to be split, and so on and so forth. Now, this growth process, like I said, happens during all three of these phases of the cell cycle, but chromosomes only duplicate during the S phase. Okay, let's hash out some details here, starting with G1. It consists of most of a cell's life and does not involve DNA synthesis, right? Instead, it consists of kind of the usual cell functions like communication and protein manufacturing, pretty much anything involved with metabolism, right? Now, let's come up with an example here. How many chromosomes are we going to start out with? Let's make it simple. Let's start out with just 4 chromosomes. Okay. Now, what do we call it when we have DNA and the proteins together? What is that structure called? Right, chromatin. Now, at the same point, we also have something called centrosomes, which we'll be using later on during mitosis or M phase. Okay. Now, how many chromosomes do we have here? Well, in total, we have 4, 2 from mom and 2 from dad. More specifically, 1 long chromosome and 1 short chromosome from mom, and 1 long chromosome and 1 short one from dad. So that means we have 2 copies of each chromosome, 2 long chromosomes, 2 short chromosomes. So are we haploid, or N, or diploid, 2N? What do you guys think? Right, that means we're 2N here, which means our ploidy is diploid. So we want to try to keep track of ploidy throughout this whole process. Ploidy process. It sounds like more P words, right? Anyway, so we're going to keep track of the ploidy throughout this whole process in the top right of our slide, up here. Oh, and by the way, organisms with a ploidy of 2n equal 4, or 2n equals 4, pretty small, pretty simple it seems. Though we did talk about that, didn't we? Anyway, so they exist. Spider mites are an example. So you might actually come across some spider mites if you do gardening at all. They tend to live on the underside of a leaf and feed off of plants. Now, they're super duper tiny small, like this little big. You'll probably see them as little red specks. But yeah, they exist. And their ploidy is 2n equals 4. Okay. next stage here, onto S phase, where we actually do the duplication of our chromosomes. So we take our long chromosome from mom and we double it, right? Making two exact copies right next to each other. We call these identical copies, what was that again? Yeah, sister chromatids. Now, these sister chromatids are actually held together at the centromere by cohesion proteins, which are going to be helpful later on when we move stuff around. Speaking of that, during the S phase, we also develop some handy components for later on, specifically kinetochores, which are these proteins that grow out from the centromere and act as like handles, right? So they're kind of removing these chromatids around. Okay. Quick note, so these are sister chromatids, but this is still considered a chromosome together, okay? A little bit confusing of how we use the word chromosome sometimes, but always stick with I like the book analogy, right? It's still the same copy, or the same version of book 1 of Harry Potter. Okay. Now, finally, in G2, that centrosome we had in G1 duplicates, so we end up with more of them. In each centrosome, we actually find 2 centrioles, which are these kind of subcomponents here. Eventually, these are used to actually move the chromosomes around. So you can kind of think of them like, think of the centrosomes as like spring-loaded spools of wire, and the centrioles are the thing that makes those wires. Okay. Now there we go, there's the centrosome. And the centrioles. Okay. All right. Now we're on to M phase, or mitotic phase of the cell cycle. The eukaryotic cell division, also known as mitosis, is broken up into 4 steps: prophase, metaphase, anaphase, and telophase. There's also another process called cytokinesis that kind of happens at the same time as telophase, and we'll talk about that later. So a handy way to kind of remember prophase, metaphase, anaphase, and telophase is you can say PMAT. Okay? Oh, that should say 4 phases, not 5 phases. It's 4. Whatever. Anyway. Okay, so the book actually has a pretty good summary of this whole process. Definitely worth taking a look at. Notice here they have a pro-metaphase. So that's actually, you could consider that a 5th phase, but we cut that out. We simplified things down. Just stick with the 4: prophase, metaphase, anaphase, telophase. Ignore the pro-metaphase. We kind of roll that in together with prophase. Besides the point. Now, remember, always stick to lecture content. That's your number 1 go-to. Okay. Now in prophase, the chromosomes start to condense. So this makes them easier to move around a little bit. They also start to become visible at this point because they're becoming more dense and closer together. It's easier to see them. Then, like the mitotic spindles or microtubules start to form from those centrosomes that we talked about earlier. These are kind of like those guide cables that are actually going to get used to pull and maneuver the chromosomes around. The centrosomes also move to the opposite sides of the cell, getting in the right position to be able to separate the sister chromatids properly. Now the nuclear envelope also breaks down at this point, which is good because that would be pretty obnoxious to have to deal with that kind of mega membrane that's hanging around in the middle with all the chromosomes inside, right? Now, finally, those spindle fibers, the kind of cables, they actually attach to the kinetochore handles that grew from the centromeres a couple of stages ago. Do you guys remember what stage that happened in? Which stage did we see the kinetochores? Anyone? There you go. Anyway. Okay. Now we're on to metaphase. Metaphase is kind of the longest stage from a time perspective during mitosis, okay? So it's the longest phase of mitosis. The centrosomes are now at complete opposite ends of the cell, and the chromosomes align along this metaphase plate, which is kind of the center line of the cell here. Notice the orientation of the sister chromatids now. So with kind of each side, one aligned with each side of the cell. For each pair of sister chromatids, one of the chromatids has a microtubule attached from one chromosome, or I should say from one centrosome, excuse me. So one of the chromatids has a microtubule attached from one of the centrosomes, while the other sister chromatid has it attached to the other centrosome. Okay. So anaphase is the shortest stage of mitosis, and it consists of the cohesion proteins that were holding the sister chromatids together getting cleaved by an enzyme. Now, if the enzyme doesn't actually cleave those proteins, we call that non-disjunction. More on that later. Now, the chromosomes are then pulled towards opposite sides of the cell. Notice at this point, we have two complete sets of chromosomes, one on each side. So this side has one full set of two chromosomes of each kind of chromosome, right? So two long chromosomes and two short chromosomes. Okay. Finally, during telophase, two new daughter cells form as the nuclear envelopes reform and a nucleoli reappears. If you guys remember what those are. So nuclear envelopes, kind of the bubble around the whole nucleus, that double membrane thing. And the nucleoli is the kind of dense portion of the nucleus. Now, concurrent to this process is something called cytokinesis, where the cytoplasm actually gets split between the two daughter cells. So let's talk about that in a little more detail here. So cytokinesis is actually not technically a step in mitosis. It's simply a process that happens concurrently to mitosis. During cytokinesis in animal cells, a cleavage furrow is formed, which is kind of this indent here, where the phospholipids are pinching inwards. Now, plants have something a little different. They get something called a cell plate that develops, that's made of rigid cellulose that forms between the two daughter cells. Now, in either of these cases, whether it's an animal and it has a cleavage furrow, whether it's a plant and has a cell plate, each new daughter cell has a full set of chromosomes identical to the original cell, as well as, of course, a full set of organelles. So that's pretty neat. So what do you think happens next now that we have these two daughter cells? What's the next step here? Any guesses? That's right, we return to the beginning. So this is a cycle. So we return back to the beginning of interphase and G1. Right, this is a cell cycle for a reason. Okay. Now, quick kind of key points or summary here about mitosis. Number one, you have identical copies are made. Number two, your goal is to make two daughter cells with the exact same genetic code as the parents. Finally, ploidy stays the same throughout this whole process. So if you start at haploid, you end as haploid. You start as diploid, you end as diploid. You start as 2n equals 4, you end as 2n equals 4. You start as n equals 100, you end as n equals 100, and so on. Okay. Next lecture, we're going to talk about meiosis and its involvement in heredity and life cycles, which we'll also end up talking about eventually how some processes that change ploidy, which will be interesting. So we'll talk a bit about heredity, including asexual reproduction and sexual reproduction, as well as how chromosomes are involved in those processes. Okay, before we get to lecture 11.1 though, let's do lecture question 10.2, sequence the cell cycle. There you go. Have at it. And see you next lecture, lecture 11.1.